Welcome back to UCrack Academy. Today we are going to see about Chapter 5 Principles of Inheritance and Variations. So I have attached two figures. You people comes to know what is going to see today in detail. For example, if you take elephant gives the birth of baby elephant. Even the mango tree if you take, அது கூட பார்த்தீங்கன்னா மாங்காய் கொட்டையை போட்டுட்டு மாங்காய் கொட்டையிலேருந்து செடி வரும், செடியிலேருந்து மரமாகும், மரத்துலேருந்து நம்மளுக்கு பழம் தான் கொடுக்கும். But it will not be changed anyway. அதே தான் வந்து human being also gives the birth of baby. So this is called a inheritance. We'll see first principles of inheritance and variations, genetics, inheritance and variations. These are three words you're supposed to know first. Always the genetics always deals with the inheritance. Variations, it is all the characters from the parents to offspring. நம்ம parents கிட்ட இருந்து தான் நம்மளுக்கு வரும். Inheritance, this is actually a process characters are passed on from parent to progeny. Like variations, it's like a degree by which progeny differed from parents. Like Mendel's law of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years, 1856 to 1863, and proposed a law of inheritance. Like Mendel conducted artificial pollination, cross-pollination experiments using several true breeding pea lines. Like a true breeding line is one that having undergo continuous self-pollination for several generations. Mendel selected 14 true breeding pea plant variations as pairs were the similar to the one character of the contrasting cat traits. Look at here dominant and recessive. இப்போ டாமினென்ட்ல பார்த்தீங்கன்னா அதோட ஹைட்டு, சைஸு, ஷேப்பு, கலரு, கோட்டு, அதோட பாட் ஷேப்பு, பாட் கலரு, ஃப்ளவரோட பொசிஷன் எல்லாமே பார்த்தீங்கன்னா டிஃப்ரெண்ட்டாக இருக்கும். ரெசிசிவில் பார்த்தீங்கன்னா அதுக்கு எல்லாமே வந்து ஆப்போசிட்டாக இருக்கிற மாதிரி இருக்கும். பார்த்தீங்கன்னா ஃபர்ஸ்ட் பார்த்தீங்கன்னா டால் இது ஷார்ட், ரவுண்டு wrinkle, எல்லோ க்ரீன் ஒயிட், இது வந்து இன்ஃப்ளாட்டட் ஃபுல்லு, இது வந்து கன்ஸ்ட்ரக்டட் ஃப்ளாட், இது வந்து க்ரீனு எல்லோ, ஆக்சைல் அண்ட் டெர்மினல். இன்ஹெரிட்டன்ஸ் ஆஃப் ஒன் ஜீன், ஒரே ஜீனில் இன்ஹெரிட்டன்ஸ் நடக்கும்போது ஃபர்ஸ்ட் இஃப் யூ டேக் கேபிட்டல் டிடி அண்ட் தென் ஸ்மால் டிடி. So this box we can say Punnett box square. This is Punnett square. Okay, TT that box. இப்போ இதுலேருந்து என்ன கேமெட் எடுத்துருக்கணும் T, இதுலேருந்து வந்து பார்த்தீங்கன்னா ஸ்மால். ஸோ F1 ஜெனரேஷன் பார்த்தீங்கன்னா எக்ஸ் இருக்கும், அதுலேருந்து கேமெட்ஸ் பார்த்தீங்கன்னா கேபிட்டல் T அண்ட் தென் இதை க்ராஸ் பண்ணோம்னா கேபிட்டல் T ஸ்மால் டி கேபிட்டல் T, ரெண்டு ஸ்மால் டி. ஸோ இப்போ எல்லாத்துலேயும் எதுலெல்லாம் கேபிட்டல் டி வந்திருக்கோ அது எல்லாமே டால் ஃபீனோடைப்புன்னு சொல்லுவாங்க. எது வந்து ஆஷ் கலரில் இருக்கோ அது வந்து ஃபுல்லாகவே ஆஷாக இருக்கும், அப்போ அது என்னது ஷார்ட். ஸோ இது ரெண்டுத்தையும் பார்த்தீங்கன்னா இதோட ரேஷியோ 3:1. திஸ் இஸ் ஒன் ஜீன். Mendel's proposition. Mendel proposed something we being stably passed down unchanged from the parent to the offspring through gametes. So over successive generation, he called these things as factors. Nowadays, we call them as that is gene. நம்ம எது ஒன்று பண்ணாலும் அது அவங்க அப்பா மாதிரி ஜீன் அவன் அப்படி தான் பண்ணுவான் அப்படின்னு சொல்கிறாங்களே, that is called proposition. Gene is therefore the unit of inheritance. The gene which codes of a pair of contrasting traits are known as alleles. That is, they are slightly different forms of the same gene. Alphabets used. Capital letters used for the traits expressed at the stage F1. Small alphabets for the other traits. Capital T is used for tall, small t is used for tract. Capital T and small t are alleles of each other. Hence in the plants, the pairs of alleles for height would be capital T, small t or rendered small t. That is what that Punnett square we have seen, no? That is what. In a true hybrid tall of draft peas varies the allelic pair of gene. For the height identical or homozygous TT and TT respectively. Capital T and small t are called the genotype. Capital T small t plant is the heterozygous for gene controlling one character height. Descriptive term tall and draft are the phenotype. Test cross. When F1 hybrids is crossed back with the recessive parent, it is known as test cross. It is used to know the genotype of the given plant animal. Look at this, homozygous, and this is also homozygous. Can you see the flowers of two? And then they have made it like a Punnett square. So with the square you comes to know what is that. Here dominant phenotype, that is genotype known. All the results are violet here. Here half of the violet, half of the white. Unknown flower is homozygous. Here also heterozygous. Law of dominance. Characters are controlled by discrete unit called factor. Factors occurs in pairs. Like in a dissimilar pair of factors, one member of a pair dominates the other recessive. Law of segregation. How they are dividing here. The alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. The parent contains two alleles and during germination or formation, gamete formation, the factors, the pair of segregations are separate from each other such that a gamete receives only one of the two factor. Like a homozygous parent produce all gametes that are similar to contain the same type of alleles. Heterozygous parent produce two kind of gametes, each having a one allele with equal proportion. Incomplete dominance. When a cross between two pure breed is done, like a F1 hybrid phenotype does not resembles either both two parents was in the between the two incomplete dominance. Inheritance of flower in the dog flowers like a snapdragon or antirrhinum species is a good example of incomplete dominance. So F2 generation phenotypes ratio 1:2:1 instead of 3:1 as a Mendelian monohybrid cross. Genotype ratio of F2 generation is 1:2:1. Look at this. Again you are going to see a Punnett square, but it is a one gene only. But the red color variety capital or white color variety here small also, that is a parent. If you take a gametes, you will get a first generation pink color. The selfing in the sense, no, you can male and female, you can do the Punnett square, but it is a incomplete dominance. So that is the F2 generation here. That is a good examples. Codominance. F1 resembles either of the two parent complete dominance. F2 offspring or F1 offspring was in between the two parents, that is also incomplete dominance. F1 generation resembles both parents side by side is a codominance. Best examples of codominance is the ABO blood grouping in human. ABO blood grouping is controlled by the gene I. The plasma membrane of the RBC, red blood cells, which means no antigens that protrude from the kind of sugar is controlled by the gene 1. The gene 1 has three traits ABI. The alleles A, B produce slightly different from sugar, while allele I does not produce any sugar. Each person possesses any two of the 3-1 gene alleles, like A, B are completely dominant over it. Like A, B presents together both express their own sugar because of codominance. Hence, RBC have a both A and B type sugar. Look at the dominance here, codominance. If you have a blood group A, the possible the genotype you will get the A only. If it is a B, B only. AB, or A or B. But the O, no options. You will get the same genotype. But the codominance here the same. Both group possibly A, possible A. And A, again you will get the A. But A, O, possibly you will get O. Like the B also the same. And then A also the same. Like A or AB or A or B. Otherwise no O. You will get no other option. You will get the O only. Multiple alleles. Examples of ABO blood grouping produce a good example of multiple alleles. They are more than 2-3 allele governing the same character. A single gene product may produce more than one effort. Starch synthesis in pea seeds is controlled by one gene. It has 2 allele B and B. Starch is synthesized effectively BB homozygote and therefore large starch produce. BB homozygous has a less effectively hence produce smaller grains. After maturation of the seed, BB round, small BB wrinkle that we have seen. Heterozygous BB, there will be a two different thing dominant, but the starch grains produced are of intermediate size. If starch grain size is considered as the phenotype, like it's a incomplete dominance. Inheritance of two genes. Law of independent assortment. When two characteristic hybrid are combined in a hybrid, which in the sense one pair of the traits, it is incomplete of the other pairs of traits. This is called the two types. Look at here, two genes. This is square of Punnett square, should be carefully listen to it. This the gametes, male and female gametes, you're supposed to memorize it. And then you can R, Y, you can. And then R, Y, R. R, Y, Y, Y. R, R, Y, Y. Small r, capital Y, capital Y. Is capital R, capital Y, small y. Like now you have to tally. Chromosomal theory of inheritance. Why Mendel theory was reminded and recognized. Firstly, communication was not easy in those days, so work is not completed properly. Secondly, the concepts of the factors or stable, discrete unit that control the expression of traits, which does not blend with each other, was not accepted by the contemporaries, and an explanation for the apparently continuous variation seen in nature. Like the mental approaches of using mathematics, biological phenomenon was totally new, unacceptable to a new biologist of that time. Finally, we could not provide any physical proof for the existence of factors. Physical நம்ம எதுவும் proof பண்ணல, அதனால we are not getting it still. Recovery of mental results. Look at here. 1990, three scientists D.V. Rees and Coherence and 1T Testmark independently rediscovered mental results on the inheritance of character. Question number 1. If a hybrid expresses a character, it is called epistatus, dominant, codominant, recessive. Answer, dominant. Question number 2. A plant having the genotype AA, capital B, small b, CC will produce kind of gametes. Options 5, 4, 3, 2. Answer 2. Question number 3. Color blindness is an dash linked recessive traits. Options: Z chromosome, Y chromosome, X chromosomes, and none of the above. Answer: X chromosome. Question number 4. In most species, mitochondrial DNA is passed down from DNA, mother and father, father, mother. Answer: mother. Question number 5. Where are the gene for cytoplasmic male sterility in plants located? Chloroplast genome, mitochondrial genome, cytoplasm, none of the above. Answer: mitochondrial genome. Question number 6. Dash is a type of traits whose phenotype is influenced by more than one gene. Monogenic traits, monogenic traits, polygenic traits, none of the above. Answer: polygenic traits. Question number 7. An individual collection of genes is called genotype, phenotype, traits, none of the above. Answer: genotype. Question number 8. A man marries a woman and both do not show any apparent traits of inheritance disease. 5 sons and 2 daughters are born, and there 3 of their sons suffering from a disease. However, none of the daughter is affected. The following mode of inheritance for the disease is sex interlinked recessive, sex linked dominant, autosomal dominant, none of the above. Sex linked recessive. Question number 9. A trait that overpower and hide another trait is called overpowering traits, complex trait, recessive trait, dominant trait. Dominant trait. Why is hemophilia a disease that is more commonly seen in males? Both 2 and 3, the disease is Y-linked, the disease is X-linked, none of the above. Answer: the disease is X-linked. Question number 11. In humans, sex is determined by chromosomes. Sex chromosomes of the father, sex chromosomes of the mother, quantity of sperm in the semen, size of egg to the fertilized. Answer: sex chromosome of the father. Question number 12. If 2 alleles of a gene that both shows their effects in the phenotype of the organism is known as codominance. One allele is dominant over the other. Both alleles are independently expressed in the heterozygotes. Alleles tightly linked on the same chromosomes. Alleles that are recessive to each other. Answer: both alleles are independently expressed in the heterozygotes. Question number 13. In this classic experiment on peas, Mendel did not utilize any form of genetic engineering. Seed shape, seed color, pod length, all of the above. Pod length. Question number 14. What was the number of pairs of constructing characterizing in pea plant studied by Mendel in this experiment? 9, 5, 6, 7. Answer: 7. Question number 15. A disorder caused by the autosomal primary non-disjunction is Turner's syndrome, Down syndrome, Clifton syndrome, sickle cell anemia. Answer: Down syndrome. I hope you all can understand about this. If you have more clarification, you can follow my video, subscribe the video. Thank you so much.